So thank you so much for the organizers for inviting me today. I was given a brief to kind of introduce methods for Bayesian adaptive trials, particularly platform trials. And I think that could almost be an entire graduate course all on its own. So I'm really going to try and do it justice in 45 minutes. So hopefully just take away like the flavor of a few pieces of information about Bayesian adaptive platform trials. So yeah, as I said, as the nice introduction says, I'm a scientist at St. Hospital. I'm also the lead of the statistical analysis committee for the practical platform trial, which is in acute care and led out of Toronto. So a lot of my kind of advice or things will be kind of based on that, and the example at the end will be from that trial. So before I kind of dive into, I guess, Bayesian analysis for adaptive platform trials, I wanted to just start with a kind of introduction to Bayesian analysis. So some of you will be familiar, some of you, it may even be the first time that you've kind of seen this idea, but essentially Bayesian analysis is a combination of kind of two key pieces of information. So the first piece of information that you need to do a Bayesian analysis, which is represented in the dashed line on the screen here, is something called a prior distribution. So a prior distribution is what you believe about the treatment effect or the quantity of interest before you start to look at the data. So in some cases, you can think about a prior distribution as just being very, very what we call vague, which basically means that you know nothing before you look at the data. Or sometimes, and this is something that we use maybe more in pediatrics, is where you might say, oh, I did a similar study in adults, and I'm going to use that information as a prior information for my belief about the treatment effect before I embark on my trial in pediatrics. So in this particular case, my prior distribution, I'm estimating a probability, and I'm saying that I think that probability is somewhere between 0.1 and 0.8. So really, I don't have any idea, but I'm giving a little bit. I know it's not really, really large. The next thing you need, and this is actually the same as you would need in a sort of standard frequentist analysis, is something which we call the likelihood, or the kind of basically what comes from the data. So this blue line here represents what we know from the data. So we collected some data, and we found that actually in this data, the kind of density or our beliefs based on the data alone say that actually our probability is somewhere between 0.8 and 0.4. So we haven't collected that much data in this example. We're still quite uncertain, but we have some information from the data. And then what a Bayesian analysis does is it creates something called a posterior distribution, which essentially combines the information from our prior beliefs and our data. So in this particular case, you can see the combination in the pink line essentially almost entirely represents the data. And that's because we had very little prior information, but it does slightly shift to the left to account for the fact that our prior distribution started out a little bit lower than our data ended up. And it also is more tight. It has more precision because we're combining the information from the prior and the data. So this essentially is Bayesian analysis in a nutshell. I think there's probably a lot of technicalities at each stage. How do you define the prior? How do you analyze the data? How do you combine them together? But this is what Bayesian analysis does. And really what we're looking for is this posterior distribution here summarizes everything we know about the quantity of interest. So if the quantity of interest is the treatment effect, that posterior distribution tells us everything we need to know about the treatment effect. So what do we do with this? So we can do lots of different things with this, but the key kind of, I think, if we take anything away from this, the posterior distribution contains all the parameter information. So we can summarize that information by taking the posterior mean. So the posterior mean would be kind of equivalent to a standard summary measure that you might get in a frequentist analysis. It tells us what the most likely value is for the odds ratio in this case. You can calculate a posterior median. So again, that's sort of a central summary measure. And it's a little bit up for debate whether the mean or the median is the most appropriate summary. But in this particular example, they're pretty similar to each other, 1.7, 1.8. So what we're seeing is we have an odds ratio, a sort of positive odds ratio from our analysis, and that's the central estimate. We can calculate the variance of this distribution or the standard deviation of this distribution. And that would be equivalent to the standard error of an estimator in a frequentist analysis. So again, that gives us some idea about our variability of our knowledge about that odds ratio. You can calculate what we call credible intervals. And a credible interval is essentially this black line on the plot here. And it tells you the kind of most likely values for the odds ratio. So in this particular case, we've calculated a 95% credible interval, which means that there's a 95% probability that the odds ratio is between 0.94 and 2.56. And what I think potentially for those of you who are used to like thinking a lot about statistics, we're often told that we're not allowed to say that a 95% confidence interval has anything to do with the probability that the true estimate is within that interval. But actually in a Bayesian analysis, that's exactly what we're allowed to say. We're allowed to say the probability that the odds ratio lies between 0.94 and 2.56 is 95%. So if we're thinking about maybe aligning this with like I guess like a statistical test in some way, we want to know what's the probability that the odds ratio is greater than 1. We can calculate that from our posterior distribution. And we can also say that because the 95% credible interval contains 1, we maybe don't have enough evidence that the odds ratio differs from 1. So in this particular case, we might say we've got a really wide estimate for our treatment effect, somewhere between 0.94 and 2.56. That's kind of how we conclude from a Bayesian analysis. So in this sense, we have a lot of kind of similar tools or similar presentations to what we might have in a standard frequentist analysis, but we do have a slightly different interpretation that we're allowed to talk about probabilities of the treatment effect being equal to or greater than 1. And this is exactly what I kind of go on to say here. So we can calculate the probability of a positive treatment effect or a something that increases the odds of something. And in this case, we have a 98% chance based on this posterior distribution that the treatment effect is greater than 1. And again, you can use that how you want. So that may be something clinically, someone might say, oh, I'm happy with 98%. Like that sounds quite high to me. And another person might say, no, there's a really bad side effect profile for this drug. I really want to be 99.5% sure that it's greater than 1. And you can think about that. You can add your kind of personalized interpretation there. So there's some kind of other key concepts. I think that that's kind of one key thing that you can do with Bayesian analysis. You can get these probabilities. Another thing that you can do with Bayesian analysis is something called a predictive distribution. So rather than a posterior, which summarizes your information about the parameter or the treatment effect, a predictive distribution summarizes information about what you think future data might look like. So that can be a really important tool when you're designing or where you're thinking about adaptive analyses in clinical trials, because you can take, this is my belief about the parameter, and if I was to collect 100 more patients, what do I think those patients would look like? Or what do I expect those patients to look like? So we have a kind of representation of that here. So we start with the trial data. So this is the treatment effect based on the trial data that we've collected. And then from this 75 patient point or 100 maybe, we're going to predict forward what we think the data might look like in the future. So this is sort of 50, just for ease of demonstration, 50 sort of potential trajectories of the treatment effect based on our current information about the parameter. And here we have a kind of zone of statistical significance. And so what you can do with a predictive distribution is you can say, what's the probability that we're going to have statistical significance if we carry on this trial into the future? So you can think about kind of two things. One is we have posterior probability, that's about the treatment effect, like what is the value of the treatment effect, or predictive probability, which is what is the probability that a future trial would behave in a certain way. So we have one that's about the treatment effect and one that's about the data. So I think in terms of trial design and trial analysis with Bayesian methods, those are really the two key concepts, this posterior distribution about the treatment effect and this predictive distribution about potential data that we might collect in our study. Cool. So I guess taking into account those kind of two key concepts in terms of analysis or terms of things that we might do, I'm now going to just move on to a couple of different Bayesian tools that we have in our machinery that can help us maybe get some additional power or some additional flexibility from a Bayesian analysis. So one thing that we can do, which is something that is used a lot in platform trials, is this concept of dynamic borrowing. So if you could look on the left-hand side of the screen, we have an example where we have 4 different states in our platform trial. So that might be different countries, that might be adolescents, children, adults, and the elderly. I don't know, I split that into 2 small things. That might be in the trial that I'm working in, it's severity states of the disease. So we have kind of non-invasive ventilation, invasive ventilation, and ECMO as the kind of severity states of our disease. And a traditional analysis would either say on the right-hand side of the screen that all these states are completely equivalent and we're going to analyze all of these data together. Or you might say to your clinical colleagues, or I might say to my clinical colleagues that actually we think that children are sufficiently different from adults that we really need to analyze these data completely separately from each other. So that would be essentially this. On the right-hand side, it's equivalent to having one trial where we throw everybody in the same bucket. And on the left-hand side, it's equivalent to having 4 completely separate trials that don't talk to each other. And one of the advantages of platform trials is that you can kind of land somewhere in the middle. So you're using all this machinery to kind of facilitate recruitment and collect the same outcomes with the same process. And what you might find is that if a treatment is effective in children, it's likely to be effective in adults. So you want to sort of borrow that information. We know that it's been effective in adults where we have thousands of recruited patients. We think that that means it's likely to be effective in children where we maybe have 200 or 300. So we do something called borrowing, which is essentially somewhere between these 2 categories, where we allow ourselves to have a different treatment effect for each state, but we're going to borrow information. So if it's effective in state 2, that makes it more likely to be effective in states 1, 3, and 4. So essentially these blue boxes in the middle. And why it's dynamic is that we basically learn from the data. So if it seems that the treatment effect in children is really, really different, we'll end up very close to this end of the spectrum. And if it looks like everything's going well, everything's basically the same across children and adults, we'll end up at this end of the spectrum. And the Bayesian analysis allows you to put a prior distribution, so basically a prior belief about where we think we are on this spectrum between independent and fully pooled analyses, and then learn from the data. So we'll have a posterior distribution about how similar our treatment effects are. So what's really nice about that is it can gain you some power in your platform trials, and it also allows you to leverage what you already have available, which is this great kind of structure where you're recruiting across all these different states, you have all this different information, and then statistically you can make the best use of that information. So it's a really nice tool that, again, is something people have seen and is talked about a lot in the world of platform trials. Oh, sorry. So there we go. We have a posterior distribution, which summarizes information about the treatment effect, a predictive distribution that summarizes information about the data, and a kind of concept to allow us to borrow information across similar but distinct subgroups. So I would say in terms of concepts of Bayesian analysis in platform trials, those are really the 3 key things that I think I see come up again and again in terms of what we're using and tools that we're using. So I wanted to talk now and bring it into the concept of Bayesian adaptive trials. Now, I actually think, Bayesian adaptive trials, frequentist adaptive trials, we have the same toolbox. We're not doing anything drastically different between the 2 types of adaptive trials. It's just the machinery that we're using to kind of do some of this analysis that's different. But here are some kind of key, specifically Bayesian adaptive trials that I wanted to just pull out. So we have dose finding studies. There are kind of 2 key classes of dose finding study that I think are kind of uniquely or very Bayesian. One is called the Bayesian continual reassessment method, which just allows you to kind of adapt very quickly to the data that you have available for the different doses and find the best dose. We also have utility-driven designs that allows you to balance between efficacy or toxicity and safety in a dose-finding trial. I know that's not really the focus of today's talk, so if you're more interested in dose-finding studies, we can sort of talk about that later. There was an excellent talk earlier about response-adaptive randomization. We can definitely do that in a Bayesian context. We can also do it in a frequentist concept. We can do sequential monitoring. So that means kind of halfway through your trial or some portion of the way through your trial, looking to see whether one of the interventions is effective or shows, like overwhelming signal of efficacy. Similarly, we can look for overwhelming signals of futility or inferiority to different interventions. And we can do kind of stopping early. Again, I think this is something that's common to all platform trials. And again, we can do it in a Bayesian context. We can do it in a frequentist context. We can think about enrichment design. So that's where you're really looking for subgroups that are more likely to benefit or have a higher potential to benefit from an intervention. Again, you can do a frequentist enrichment design. You can do a Bayesian enrichment design. And we can think, which I think also comes to this platform concept, we can think about seamless designs. So where you might want to move from dose finding to phase 2 to phase 3 to phase 4 within the same platform or within the same infrastructure, and potentially even borrow or use your information from phase 2 as a prior for your phase 3 results. That's something that you can also do in a Bayesian context to really make sure that you're using or making the best use of the patients who have kindly given their time and their effort and their energy to be in the trial. So I don't think there's anything particularly kind of magic. This is a lot of concepts that we've seen in all these different types of adaptive trials, and this is something we can do in a Bayesian setting. So now, I guess, I was asked to introduce Bayesian trials and then talk a little bit about how we do sample size calculations or analysis of Bayesian trials. So I think the key challenge, opportunity, and experience of Bayesian clinical trial design is that we need to run simulations. So I always like to kind of reflect back on this FDA definition of an adaptive design. So an adaptive design is defined as a clinical trial that allows for prospectively planned modifications to one or more aspects of the design based on accumulating data from subjects in the trial. So there's two key kind of things, again, highlighted in blue here. An adaptive design means we need to pre-specify or prospectively decide what adaptations we're going to do. And I think there was a good highlight that maybe the recovery trial in its speed didn't quite do that as well as it could do. But what we really want to do is we want to plan, prospectively state what are going to be the modifications of our trial. And it's going to be something that's modified based on the data that we collect in the study. So it's not about saying, oh, we're just going to add a treatment arm in 10 months' time. There is an adaptation, but really the FDA says that it needs to be in response to the data that you're collecting. So the goal of statistical trial design, or in this particular talk, Bayesian trial design, is to really determine which adaptations should be made. So, what are going to be the rules for adapting your trial based on the data? And then secondly, the impact of these adaptations on the kind of statistical properties of the trial itself. So we want to make sure, again, we spoke a little bit about maybe not having adequate control of type 1 error. Statistical trial design sort of aims to make sure or to demonstrate or prove that we have good control of these statistical properties. Do we have the ability to truly detect the difference between the interventions if it exists? So we really need to decide, pre-specify decision rules for the trial adaptations. This is how we're going to adapt our trial in response to data. And in particular, the way that I think about it is that what we're really doing is we're deciding a trigger for each adaptation. So if we were doing a dose finding design, we would determine a trigger for moving to the next dose. If we're doing a futility analysis, we will determine the trigger that decides which intervention is futile. If we're doing an enrichment design, we will decide the trigger for moving into a specific subgroup instead of staying in the whole population. So that's really what we're doing. We're setting out a statistical rule that's going to trigger our change in the design of the trial. And particularly in Bayesian analysis, simulations are usually required. I actually have never done a design that didn't require simulations from a Bayesian perspective. There probably are, I'm sure there are some people in the room who may be able to tell me of one or two, but it's almost always a simulation-based design. So I was asked to talk about how to design a trial and specifically how to design the simulation or how to design a Bayesian adaptive trial. So I actually went back. I have a document that I give to everybody who comes into my lab, and I've actually not found something that's like published and really clearly outlines all of these things. So this is my personal idea of what I think you should do to design a trial. If someone knows of a really good paper, that would be amazing. And then I can give it to my trainees when they come in. But I always think that it starts, perhaps in all research, with specifying the research question. I need to know from the clinical team, what is the research question? Population, interventions, comparator outcome. There's nothing that you can do in a simulation-based design without knowing those key pieces of information. And additionally, I need to know the goal of the adaptive design. Now, I think it's becoming more and more common for people to want to do adaptive designs, to see the benefit, to see the value. But really, as we already showed, there's so many different types of adaptations that you could do. What are we really looking for to do in that design? And that's when I can start to think about, okay, what are the appropriate adaptations? What are the tools in my toolkit that I can use to answer the clinical reason for adaptation? So I think we don't want to complexity for complexity's sake. We really want to make sure that the complexity that we're adding is answering the clinical question. So that's really where I start, and where I encourage everybody to start. So once you have that, you have to develop your model, your statistical model for data analysis and for simulation. So those models should really kind of mirror each other, but crucially, there are two parts there. One is the data analysis that you will do once you have the trial data. And the other is what you need for the simulation, which is generating kind of fake or pseudo data in your computer to be able to run the trial lots and lots of times. They should match each other, but crucially, in order to simulate, you need some information about the expected distribution of the outcome in the trial. So you cannot do that without going back, firstly, to your PICO, what data are we interested in? What population are we interested in? And what data do we have that can help us simulate patients that look like our population and the outcome of interest? And again, my next few slides is going through an example of this. So hopefully everything will become a little bit clearer as we go through. The third thing is to design the trial decision rules. Now, sometimes you might just sort of specify your trial decision rules, and sometimes you might use your simulations to help you design the optimal or the ideal trial decision rules. And again, in the example, I'll show you a setting where we actually tried to optimize those trial decision rules. And then we need to determine the key scenarios for simulation. So, we're going to detect a treatment effect of an absolute risk reduction of 0.1, or do we want to detect an absolute risk reduction of 0.5? Those are key scenarios that we need, and they will have an impact on the power of the trial. So that's really what we want to think about. What are the clinically relevant scenarios for the treatment effect that you're expecting to see in the trial? And then number 5, run simulation. I wish this was the nice, easy bit where you're all done. Sometimes this can take seconds, sometimes it can take months. So really, it's crucially important to make sure that you've got all of this specified in stone, because you really don't want to have to go back to the drawing board and be like, oh, actually the outcome was wrong. And so you've spent your months doing the simulation, and now actually we need a binary instead of a continuous outcome. And then finally, for me, a really crucial element is evaluating the results, obviously, but then also communicating those to key stakeholders. So they may be people, almost everybody in this room, I suspect, would be the kind of key stakeholder in a clinical trial simulation presentation. But it may also be people like your patient partners in the study, in which case you will have to think really, really carefully about how you're doing that presentation. So I have about 20 minutes left, and I just want to go through a step-by-step of how I designed a real trial using those 6 key steps. So as I said, I'm the leader of the leads of the statistical analysis committee for this practical platform trial. So that's what I'm going to use as an example in this particular presentation. I haven't listed everybody who's involved in the practical platform trial, but make it be known that it is a lot of people, and this is not my personal work that I've done all on my own, and I just want to acknowledge that. So again, we start with a PICO. So in this particular case, our population is patients with acute hypoxemic respiratory failure. So essentially, we are in the ICU. These patients are having respiratory failure. They're struggling to breathe, and they have some sort of intervention to support that. Some might be non-invasive ventilation, so fairly low intensity, all the way up to ECMO. The intervention in this particular setting is a novel ventilation strategy. So it's for people who are on the ventilator, having the ventilator breathe for them, and we're developing, or my amazing clinical colleagues have developed a novel strategy for ventilating those patients. And then we're comparing it to standard of care, so standard ventilator settings. My clinical knowledge is not as good as it could be, so that's my understanding. And in this particular case, our outcome is ventilator-free days. So essentially, this is the number of days that the patient spends not on the ventilator after randomization into the study, up to day 28. And patients who die during the first 28 days are given the worst possible outcome on the scale of -1. So essentially, it's a composite outcome of death and length of time on the ventilator. And we have, I guess, like a factorial design. So this is what's represented here. We have these 4 severity states that I showed before, where this is the least severe and this is the most severe. And then at the moment, we have 3 funded trials kind of within this platform. But I'm just going to focus on this one here that only focuses on those 2 internal severity states. So essentially, we have this 3rd trial here. We have 2 severity states where we're going to be estimating separate treatment effects within each state. And the adaptations that we're going to think of are futility. So is the novel intervention not good enough, essentially? Like, this is a more complex intervention to use in clinical practice. So we want to make sure not just that it's effective, but that it's effective enough to encourage the switch from the standard of care. Efficacy, so is the new intervention effective? Does it work? And then we're also going to account for our treatment effect heterogeneity between the 2 different severity states. So those are our kind of key Bayesian tools that we're going to bring in. So we needed to think of a model for the data. And I believe that John was using a similar outcome earlier. So we essentially have as I said, death is equal to -1. So here we actually have a very high rate of death in this population of, I want to say, about 37%. So really that death spike is quite severe. And then what we have is a much flatter distribution where people who survive tend to be liberated from the ventilator fairly early on in their clinical course. Although there are some patients who remain on the ventilator at day 28. So this is a 0. So for those who are used to looking at nice normal distributions to do all of their analysis, you'll see that this distribution pretty much brings fear into the heart of any statistician who's looking at not just kind of peaked data, a nice spread in the middle, another peak over here. We have a lot of challenges with this data. But the way that we've chosen to work with it is to think about simulating the number of patients in each category. So we're really treating this as a discrete distribution in which we just calculate the proportion of people in each category. And the treatment effect is going to be estimated with a proportional odds model. So it does give you an odds ratio, but it's the odds of moving from one category to the next. And we assume that it's all the same throughout the whole scale. So essentially, it basically is the odds of moving from a worse category to a better category. So something that changes you, that pushes you up this scale, is a positive treatment effect. And again, to account for heterogeneity across the groups, we're going to use this dynamic borrowing concept. So we have these two groups. We're going to have separate treatment effects in each group, but if they're similar, we're going to bring them together. So what are our trial decision rules? Again, we spoke kind of already, I guess, about this. We have a stopping rule for futility and a stopping rule for superiority. So in this particular case, I have a kind of depiction of this. We have the treatment effect as the trial enrolls. We have the 95% credible interval of the treatment effect as the trial enrolls. So what you can see is that as we get more patients, the interval actually becomes shorter and shorter. So we're getting more information about the treatment effect, exactly as we'd expect. And what we're looking for is the treatment effect to either fall essentially wholly within this superiority region, which would indicate a positive treatment effect, or to fall in this futility region. And because we're more conservative about, I guess, type 1 error incorrectly concluding superiority, we have a higher threshold for falling in the superiority region than we do in the futility region. So in this case, our trial stopped actually before the 95% credible interval fell within this, because we need only a 90% probability of futility to stop the trial. The two severity states will be evaluated separately. So we'll have one plot for the low severity and one plot for the high severity, and we'll have analyses every 3 months. So we're really thinking about a kind of highly adaptive trial once we get past an initial recruitment. So what did we think about here? So the first piece of the puzzle is to determine the thresholds for making decisions. So I said, we need to have these triggers. We need to know exactly when we're going to adapt the trial, stop for futility, stop for efficacy. And in order to do that, we want to make sure that our trial has good performance. It has good type 1 error, it has high power, it has low sample size, all of the things that you would see in a traditional trial design. And the way that we did that is we actually had 128 different scenarios where we varied the probability threshold to stop for superiority. So you can see these are very high values where we're looking to say we're really, really sure that this treatment is superior before we go forward and we say, yep, superiority. We also have probability thresholds to stop for futility. And again, these are much lower. So we're really looking for like a less conclusive signal for futility. We have an initial recruitment in our trial. So we want to make sure that we're not looking at the data when we've got 10 patients in the trial and being like, yeah, great, we're done. We've absolutely nailed this. So we started with 200, 350, 500, and 800. And again, this is really where the statistics and the clinical really come together. Like probably statistically, we could look a little bit earlier than that, but the clinical team were like, no one's going to believe a trial result based on 200 patients. Like there's no point. So that's really where we started some of those thresholds. And then we just looked at 2 odds ratios. So we looked at an odds ratio of 1, which represents no effect. So that's really where we're looking at that type 1 error. And that's where we want to make sure we've got good control of that. And then 1.3 was the odds ratio that we chose to evaluate power just in this scenario. So to give you an idea, in this scenario, we're just trying to maximize power at odds ratio of 1.3 for a minimal sample size. So that's what we chose. And that, I think, again, came from the clinical team. That's something that they're expecting to see. I think, yeah, that's the hope, I think. And then once we selected our design, we wanted to dive a little bit more deeply into this. So obviously an odds ratio of 1.3 is what we hope, but there's a potential that it could be any range of different things. So we really wanted to evaluate the performance of the design in lots of different scenarios. And in particular, one setting that was really important to the clinical team was the harm scenario. What if this novel ventilation doesn't work at all and we actually harm people? What's our design going to do in that case? And what you'll actually see in our simulations is that it's really good. It just cuts out really, really quickly. So that was something that was comforting to the clinical team. And then really we're looking for this range of different scenarios, all the way to like probably the highest benefit that anyone's ever seen in a critical care trial, down to some of these more like equivocal analyses. And we just wanted to see, you know, are we going to waste time? How many patients are we going to have to recruit in these settings? And the other crucial set of scenarios that were really important was we have this idea of borrowing. We're going to try and use information across the two groups if they're the same as each other. And so we started by doing all these scenarios where they were the same. And we were like, oh, we're doing so well with our borrowing. This is great. It's really working. And then we thought, oh, actually, you know what? What if the effects are different from each other? How is that borrowing going to affect us? So in particular, when one state has no effect and the other state has a big effect or some effect, how does that affect the type 1 error in the state that doesn't have an effect? So again, this is a lot of kind of things to think about. And crucially, you don't want to have to go back to the drawing board and run these again. If you're running 128 scenarios, you don't want to be thinking, oh, we chose the wrong outcome or the distribution of the underlying effect is wrong. So really, this is where steps 1 to 3 are really, really crucial to come about. So I couldn't give you a slide for number 5. I could just sort of stand here in silence for a few minutes while I pretend to run the simulations. But really, the crucial piece of information is really how to present the results. And it's certainly not an easy task. This is one way that we've come up with doing it. So this is the 128 design scenarios. So these are the results trying to pick the best design for our trial. And just to orientate yourself on these plots, on the x-axis, we have the expected sample size of the trial. And on the y-axis, we have the power. So if you're thinking about your traditional way of designing trials, we really want to be here. We want to have really high power and really low expected sample size. But of course, you can sort of see that there's like a correlation. The higher the expected sample size, the higher the power, which at least when we got these results was comforting to be like, yeah, this is probably right because we're pretty sure that we're in the right ballpark. The crucial thing to note here is that we've taken off all of the designs that had a type 1 error greater than 5%. So we would potentially have some designs that are really, really great up here, but they have really poor control of type 1 error. Type 1 error is 20% because that makes it really easy to detect an effect. So really, these are the designs that sort of allow us to have good control of type 1 error, but have sample size and power. So really what we're looking for is we're looking for designs on the left-hand side of this plot. So, and this is the same design across the two plots. So you can see this like small triangle here is on both plots. So we're looking for ones that are on the left-hand side in both plots. Perfect. And you can see that as the initial recruitment increases, the power increases, but the sample size increases. You can see that as the superiority probability increases, the power kind of stays the same, but the sample size increases. So really, we were looking for a sort of lower superiority probability. And then for the futility probability, as the futility probability increases, the power increases, but so does the expected sample size. So we're looking for a design that sort of balances all of those things. And I believe we went for this blue square here, which is a futility probability of 0.995. So really, really certain. Sorry, a superiority probability of 995, futility of 95, and an initial recruitment of 350. And then finally, to give you the simulation results for the different odds ratios. Again, these are very difficult to present, and I'll just give you a little snapshot of what we did. But this is an increase in trials. As the trial sample size increases, what's the probability of superiority and what's the probability of futility? So what you see, this 0.8, we basically go straight at our interim and we're out. Similarly, if you have a really high odds ratio, you get the same thing for superiority. And that is the end of my slides. I think I'm pretty much on time. So thank you, and I'm happy to take any questions.